Well, a warm welcome to today's talk, Tuesday the 15th of June. Now, a little bit different today. I want to look at a new vaccine, the Novavax vaccine as reported. And there's also a fascinating trial from Israel looking at Ivermectin, which we've looked at before. So the first part of this video, we're going to look at the Novavax vaccine. New vaccine, very encouraging looking. And the Ivermectin report from Israel, which is also very encouraging. So hopefully an encouraging video today. Now, the Novavax information, it's not a peer-reviewed paper yet. It will be peer-reviewed in time. But at the moment, it's news by press release. And of course, we had this with all the vaccines. So this is from the Novavax site. We believe that the data has been reviewed by independent panels. But this is how the information is initially given out, and that came out on the 14th of June. So that just came out yesterday. And it is a very interesting looking vaccine. Now, it's a protein-based vaccine. It's made in the same way as, for example, some influenza vaccines are made this way. And HPV, the human papillomavirus vaccine, is made this way. Human papillomavirus is the virus that causes nearly all cases of cervical cancer. So let's look at that now. So it came out on the 14th of June. That's the code for their vaccine there. That is the link there that I've just shown you, which is that one. So basically, this is what we know about it at the moment. So, but it is fairly comprehensive. And in the past, when we've looked at these reports from other vaccines, they have turned out to be essentially consistent with what was later published in peer-reviewed publications. It's reporting on the phase 3 trial. Now, it's a recombinant nanoparticle, so it's a very small particle. It's recombinant, so it's a protein that's been made up, and it's also good because I think I've mentioned that lower down, but you can keep it in the fridge. It's 2 doses, 3 weeks apart, and it can be stored in the refrigerator temperatures. So what actually happens here is the information to make the protein, the genetic information to make the protein, is actually put inside another vector virus, but that goes into moth cells. Actually, moth cells. So the DNA to make the protein is put into moth cells. And these moth cells are brewed up in huge cultures, massive vats, but all very similar types or the same type of moth cells. And because the moth cell is infected with these viruses that carry the information to make the protein, and it's a spike protein that actually makes, then the moth cells just produce lots of this protein. Then it's just a matter of purifying it. So it's a protein. So, and it's basically a spike protein. So what these moth cells are doing for us is kindly making these spike proteins that we're fairly familiar with now. And groups of these spike proteins will stick together. So they have the antigenic properties of the SARS coronavirus 2. But of course, it's not the virus. It can't reproduce. But these are the antigens that the immune system recognises. So it actually injects the protein directly. So whereas the mRNA vaccines get your own body cells to make the vaccine, to make the vaccine makes your own body cells make the antigen. The messenger RNA goes in and codes for the cell to make the protein. The cell makes it. And with the adenovirus vector vaccines, the DNA from the adenovirus vector goes into the cell, and again the cell makes the protein. Well, this is not, it's passive. The whole protein is given ready-made. And fairly new technology. As I say, the only place I know it's being used is the influenza and the HPV vaccine. There might be one or two others, but they're the ones I can remember. Now, this Novavax is reported to give 100% protection against moderate and severe disease. Pretty impressive. All the hospitalizations and deaths were in the placebo group. And this is a placebo-controlled, double-blind, randomized trial. It's 2-to-1. That means twice as many people had the vaccine as had the placebo. Overall efficacy was 90.4%, and even slightly better actually in high-risk populations. So very good for high-risk populations, such as the elderly or those with comorbidities who were well protected. Good size sample, 29,960 patients were recruited. 119 sites, mostly in the United States and Mexico. So this is a proper phase 3 randomized double-blind controlled trial, no question about that. Now there was arms of the trial in other countries as well. There was an arm of the trial in the UK, but this is the information from the US and Mexico data. So they filed for regulatory authorities in the third quarter of the year. So what, July, August, September time. We would expect this to be approved fairly quickly from the information that we have at the moment. Once it's approved, they can make 100 million doses per month and 150 million doses per month by the end of 2021. So large-scale production. It's going to be made in the northeast of England. In the UK, it's going to be bottled by GlaxoSmithKline. 60 million doses a year. They're going to bottle it in Barnard Castle, also in the northeast. I think it's being made in Billingham in the northeast. So it's a short ride from where it's produced to where it's bottled in Barnard Castle. Now, this 60 million doses a year that's going to be made in the UK, I'm not sure if this is in addition to this 150 million doses. I think it probably is. So that would be 210 million doses of this being produced per year. So pretty large scale production. And looking good. Now the results are consistent in the US and Mexico. High efficacy amongst circulating variants. Now we're going to come on to the time in a minute. But anyway, there were 77 infections in the trial. 63 of those infections were in the placebo group. 10 of those 63 infections were moderate cases, 4 severe. So that was what happened in the placebo group. The vaccine group, there was only 14 infections, and all cases observed in the vaccine group were mild. So there's the difference between the two. Quite stark and significant differences, giving the high level of protection against symptomatic disease, and all cases observed in the vaccine group were mild, so giving 100% protection from this data. Okay, it's just under 30,000 people, but from this data, it's looking pretty good. Now, as I was saying, the date here, 25th of January through to the 30th of April. Now, this is relevant, of course, because this is basically pre-Delta variant times. But the Alpha variant, the UK Kent variant, the B117, became the predominant strain in the United States, to some extent in Mexico as well. So a lot of this data is against the B117. So although it's not the initial virus it was planned to protect against, that was the original wild type, it did pretty well. Will it do slightly less well against the Delta India variant? Probably, but only slightly, we would expect, based on the other vaccines. Slight reduction in efficacy perhaps, but there again we don't know that because the trial hasn't been done yet. 100% efficacy against variants not considered variants of concern. So previous variants, 100% efficiency. The variant of concern, the B117, we're looking at just over 90% efficiency. So I'd be very surprised if it wasn't at least 80% efficient against the Delta variant, and I would expect it to be more than that. Favourable safety profile, generally well tolerated. Serious and severe adverse events were few, but balanced between the vaccine and the control group, so that they were in both groups essentially the same. Infection site, pain and tenderness, generally mild to moderate, lasting for 3 days, as you would expect. Systemic side effects, fatigue, headache, muscle pain, lasting less than 2 days, but all considered acceptable, and it was generally well tolerated. Now, the placebo-controlled portion of the PREVENT-19, which is the trial, continues in adolescent groups 12 to 18 years of age. So that's being done at the moment. So far, they've enrolled 2,248 people in that age group. And this vaccine has got a lot of money behind it. It was part of Operation Warp Speed under the previous Trump administration. It was awarded $1.6 billion. That's a fantastic amount of money for 100 million future doses that paid for the development. So largely paid for by the United States taxpayers. So it's come on quite a bit later than the other ones, but it is a very nice looking vaccine from the data we have. And it may well be that this is used for third doses of vaccines, should that become necessary. We're still planning for a third dose of vaccine in the UK, perhaps in the United States as well, and this could well be the one that's used. It's looking good so far. So, of course, with disease, we want to prevent it. We can prevent disease with vaccination. We can prevent disease, in some instances, with chemoprophylaxis, using drugs as prophylaxis. If someone gets the infection, we want to be able to treat it. So far, most of the emphasis in this pandemic has been on preventing using vaccination. But ivermectin is a very promising looking treatment, reducing viral load, as we've looked at before. Now, this is important because if it reduces viral load, that's also going to reduce the transmissibility. If I've got the infection and you reduce my viral load with a drug, in this case with ivermectin, as we believe works, then that's going to stop me spreading it to you, which of course is desirable. Being used in some areas of the world, not in other parts of the world. Had it been used in Bolton, would that have made a difference to the spread of the Delta variant throughout the UK? Because ivermectin works equally well, as far as we know, equally well amongst all variants. So I'd like Dr. Chris Whitty to answer that question, please. Don't expect he will, but I'd like him to answer that question. And just before we look at the study, how many people have heard Dr. Fauci talking about ivermectin? Chris Whitty talking about ivermectin. Dr. Tedros talking about ivermectin. Sir Patrick Vallance talking about ivermectin. There's sufficient data on the efficacy of this drug, or the likely efficacy of this drug, that absolutely demands an official response, but we don't get one. When's the last time, hands up if you saw ivermectin being reported on mainstream media? Neither have I. Didn't see any hands at all. So we really have to ask why my questions and other questions are not being answered by world authorities on this, because there is sufficient data to merit an answer to that question. I'm not a regulatory authority. I'm not your prescriber. I can't say use it. But I am asking the regulatory authorities to answer this question. And so far they haven't. So this is an educational channel. It's not for you don't take my medical advice from me, of course, but the regulatory authorities and the senior doctors really should be answering this question. Come on, Tony Fauci, answer the question. Come on, Chris Whitty, answer the question. Come on, Dr. Tedros, answer the question. Even if it's no John Campbell's talking absolute rubbish, the data merits an answer. Right, enough of that. Let's go on to the study itself. Favourable outcomes on viral load and culture viability using ivermectin in early treatment of non-hospitalised patients with mild COVID-19, a double-blind, randomised, placebo-controlled trial. So that sounds pretty good to me. Carried out at the Sheba Medical Centre and in collaboration with Tel Aviv University in Israel. Now, by way of background, they say ivermectin is an FDA-approved broad-spectrum antiparasitic agent. We know that. Initially approved for human use in 1987. The discoverers of ivermectin received the Nobel Prize for Medicine, no less. The Nobel Prize for Medicine, for goodness sake, in 2015. I mean, a Nobel Prize for Medicine, does it get any better than that? You know, that's the ultimate accolade, isn't it? And the discoverers of ivermectin received the Nobel Prize for Medicine in 2015, and yet we hear nothing now from our doctors and mainstream media. Direct quote from this article: with its high profile, ivermectin is a potential treatment against COVID-19 in its different stages. Now, they mention different stages here. Here's the actual paper here. Now, word of warning here, it's a preprint, it's not peer-reviewed, but it's from reputable people in reputable institutions, and I've read it, and it looks good to me as far as I can tell from the information that we've been given in this preprint, which is quite a lot of information. So the idea of ivermectin at work in different stages is if you give it before someone's infected as a prophylactic dose, then the idea is it stops someone getting infected. It's a preventative, and that's important because that could form a firewall around infections and stop it from spreading. Pity that hasn't been done in the UK. Or let me rephrase that: the authorities should say why that hasn't been done in the UK. but of course they don't. But the evidence is there to merit them answering that question. So acts as a preventative, if it is indeed antiviral, as the evidence suggests on this paper, and in many other papers we looked at. If it acts as an antiviral, it's a treatment, because viral load seems to be proportional to the severity of the disease, to at least to some extent. And it's also anti-inflammatory, which means it might be good for later stage COVID and potentially, and I'm really hoping trials are going to start on this soon, long COVID. Long COVID. A lot of people believe it could be efficacious in long COVID, and we really do need the trial on that. So high safety profile. So this group is clearly saying it's got a high safety profile. Now, some people have tried to intimate that it's not a safe drug, but this paper would appear to disagree. This is a direct quote from this paper saying it has a high safety profile. Right, so the trial, that's the link there to the trial. To assess whether ivermectin can shorten viral shedding in patients in early stage of COVID-19 infection. Now, if it prevents viral shedding, that you would imagine would reduce the spread of the disease. And if people aren't shedding the virus, then presumably they have less of the virus in their body, therefore are likely to become less sick. Let's see if they present any evidence for this. So as we said, double-blind randomized controlled trial, brilliant. Now the dose they gave was fairly low dose, 0.2 milligrams per kilogram for 3 days. So that's the lower side of the dose. Various groups are advising a higher dose for longer than that now, but that's what they said, and that's what they did. So it's on the low side. So if given it's a relatively lowish dose, we would expect these results to be minimum results. And if a higher dose was given, who knows, the result might be better. We don't know that, of course, because we didn't test for that in this particular study. But other studies do tend to indicate exactly that. Non-hospitalised COVID-19 patients, PCR testing, of course. PCR tests at recruitment and then every 2 days to monitor the viral load. Now, the primary endpoint was the reduction of viral load on day 6. So day 1, 2 and 3 they were given the ivermectin. Days 3, 4 and 5 they were given nothing, but the result was measured on day 6 when again they were given nothing. So the drug was only given for the first 3 days. Now, reduction in viral load was taken as a CT level, cycle threshold level of greater than 30. So that symbol is greater than, isn't it? Greater than. So in other words, it was CT cycle thresholds of 31, 32, 33, 34, 35, whatever, indicating that there was a lower level of the virus there because more cycle thresholds were needed to detect it. So, of course, the greater the CT value, the lower the level of the virus. So if this got over 30, then they said that was the non-infectious level, because you had to multiply it more than 30 times to get a positive result. Of course, if someone has got a heavy infection, then they'll test positive with lower CT values. But this is the value they took here. Reflected as a CT level greater than 30, and if that is greater than 30, they classified that as essentially non-infectious for the purposes of this study. Primary outcome was supported by determination of viral culture viability as well. So what they did here as well. Now, when you want to grow up a bacteria, of course, you just put it on an agar plate and you grow it up. And it grows. But of course, viruses only reproduce inside cells. So to culture a virus is more complicated. They cultured it inside a standard cell culture line. And they saw if the virus was able to replicate inside these cells. So they still cultured the virus. But it was inside cells, not in a petri dish. That's the only difference. But they did it, so that's good. In the old days, all these it used to be done inside eggs, but now they seem to use mostly cell cultures for this. Okay, the results. 89 people in the study. Okay, we would have liked it to be bigger, but the results were still significant. Now, the ivermectin group, there was 47 in that group. The placebo group, there was 42 in that group. So that meant they could compare the results in that group with that group because it's placebo controlled. These were given tablets of the same size, colour and everything as these groups at the same time, and they didn't know whether they were getting the real tablets or not. Mostly men in this study, only 21.6 female, so that's perhaps not ideal, but nevertheless. Asymptomatic recruitment 16.8, 83% symptomatic. So mostly patients symptomatic, but some were picked up in quarantine hotels in Israel who were still asymptomatic, went on the trial. On day 6, the ivermectin group, 34 out of 47, 72% reached the endpoint. In other words, they were in the non-infectious range. Placebo group, 21 out of 42, that's 50%, reached the endpoint. In other words, they were considered non-infectious. So we see 34, 21 in the placebo group, non-infectious, 72% in the ivermectin group, 50% in the placebo group. So we see the ivermectin group, more of them became non-infectious on day 6, indicating lower viral shedding and presumably lower viral loads. The odds of a negative test on day 6 was, after they did the sums, was 6.26 higher in the ivermectin group. So better than 2 times better, not as good as 3 times better, 2.62 times higher in the ivermectin group, the odds of getting a negative test. And this, of course, is with the low dose. This is 0.2 milligrams for 3 days. So quite an impressive result, 2.6 times more likely to get a negative result if they'd taken ivermectin. 2.6 times more likely to get a negative result, which is what we want. Also, ivermectin group cultures at day 2 to 6 were positive in 3 out of 23, that's 13% positive for cell cultures. Placebo group cultures at day 2 to 6 were positive in 14 out of 29, 48.2% were positive for cultures. So the ivermectin group, only 13% could culture the virus, 48.2% in the placebo group could culture the virus. That result is a p-value of 0.008%. So there's only 8 chances in 1,000 that that result arose by chance. 3 hospitalizations, 3 in the placebo group. So 3 hospitalizations, 3 in the placebo group. So all the hospitalizations were in the placebo group. No one hospitalized in the ivermectin group. Diarrhea, 2 people in the ivermectin group had diarrhea as a side effect. 1 person in the placebo group had diarrhea as a side effect. No other adverse effects were reported. Now, this is a direct quote. No other adverse effects were reported. Direct quote from this paper. If you don't agree, there'll be an address for correspondence on there. Tell them that this is what they are saying. Conclusions: there were significantly lower viral loads and viable cultures in the ivermectin group, which could lead to shortened isolation time in these patients. Lower viral shedding, lower spread of the disease. Lower viral shedding indicates lower viral load associated with less severe disease. What's not to like? So we've given many different lines of evidence for the efficacy of ivermectin as an antiviral agent in many videos, and it merits an official response. And you really got to wonder why we don't get one. You can leave comments on that if you want. I couldn't possibly comment. Right, that is us for today. Food for thought. Now, this is really encouraging because, there is indications that ivermectin is effective against other viral infections like dengue, for example. So it really is up to governments to take this forward for the good of everyone and for the good of their populations. So message to governments there, message to all in authority. Please answer my question. Thank you very much. And of course, thank you for watching.